Hi, today we're going to discuss about physiological principles, especially cardiovascular system, circulatory system. So we want to study about the circulatory system, which is consisting of circulating fluid and system of vessels, in this case, blood vessels, and the pump system. And so we want to understand the general structure of the blood vessel first. And from more engineering point of view, we want to grasp the relationship among vessel radius and the fluid resistance and pressure drop, which is a driving force for blood flow. And the business end of our circulation is the capillaries, which is, we want to study the function of the capillaries and the distribution of fluid throughout the tissue so that we can adequately provide oxygen and nutrients and collect waste and the transport of molecules. And for this, we want to study the pump system, which is the heart. So understanding anatomy of the heart, and the heart is an electrical organ, that electrical system which generates coordinated contractions of the heart muscle. And to get into more of this, the sequence of events of this cardiac muscle, which is cyclic, and that lead to pressure generation, which is driving the fluid throughout our body and within these heart chambers and the aorta. So let me start with showing you a movie of the blood vessel. So the circulatory system. So this movie is what I take with the microscope with a mouse brain. And that red one, let me show from top to bottom. These red ones are blood vessels. You see these are arterioles, diving, penetrating arterial. And these little things, these are capillary vessels. You see how beautifully distanced with these capillaries. By the way, this scale bar is about 100 microns. So you can see how much of distance between these capillaries. And what does do this capillary is mixing, exposing cells, which is not visible here, throughout our body, in this case, brain, and oxygen and nutrients are diffused out of these capillaries. And that provides a pathway, also removal of waste products. And this movie is about 700 by 700 micron field of view, and the depth is about 650 micron. So not only this physiological, normal physiological aspect, blood vessel and circulation is so important in pathology as well. For example, in cancer, we've already talked that the cancer growth, which is a tumor forming, and the tumor cells ask for more blood vessels so that they can feed themselves. And that we call as a new vessel growth from existing vessel called angiogenesis. And recent science paper published about another aspect of to feed a tumor here called vasculargenic mimicry. Let me reiterate, there are big two ways of forming vessels. One is existing vessels and forming a new vessels, such as angiogenesis. Another way is thinking about the development. You have to create blood vessels from, not existing vessels. That we call vasculogenesis. Okay, so what people have found is in tumor, very interesting phenomena, that the tumor cells organize themselves into form something like vessel-like structures, these hollow channels inside. And then normal blood vessels hook up to channels within the tumor mass to feed the tumor. So this is named as a vasculogenic mimicry because it mimics vasculogenesis in normal physiology. And why distinguishing and finding this is important for us? Because when we are treating or killing the tumor mass, we could generate anti-angiogenic therapy, which you might develop a new antibody drug that grabs the molecules, which is growing a new vessel form. But then molecular mechanism might work different in in this vasculogenic mimicry, then the treatment has to be different. So in this regards, we have to understand various ways of looking at what's happening and the molecular mechanism and how to think of the treatment if it's a pathologic condition. While we will not know whether this vasculogenic mimicry is so general that it can stay longer of this hypothesis, but we will see in the future. So let me start with the human circulatory system. Of course, our vessels are arteries and veins. These are defined as whether the blood is coming out away from the heart is called the artery, and towards to the heart is called the veins. And this is an interesting comprehensive one I want to go through with you. We have a heart, and heart pumps out of blood, and they can go to here as a head and neck, so this part. And this arterial blood also go to arms, limbs, and also it can go to our heart, our lungs, to oxygenate the blood, so it's pulmonary. And interestingly, the heart sending out the blood, but the heart muscle is one of the most hard-working organ in our body, because 24 hours, 7 days a day, 365 days, it's always working. So that heart muscle have to be fed well by oxygenated blood from the heart. It goes back into the heart, and that's called coronary arteries. So it feeds the heart muscle as well. Let's see another part. Heart goes to our trunk, and heart arterial blood goes to our liver, called hepatic artery, and which we already discussed before. And another one goes to splenic artery, and also it goes to the portal vein. And you see, portal vein this is guts. Guts, that venous blood contain a lot of nutrients from our food, goes collected as a portal vein and go parallel with the hepatic artery in the liver. That exquisite structure make them mix together in liver sinusoid. And then those will collect into the central vein, and that drainage is called hepatic vein. So another one we haven't talked about yet, but it's called to kidneys. It's called the renal arteries, and that has specific glomeruli. And you know, from the organ, when it comes into the organ, it's called afferent. And from there outside, like an exit, is called efferent arterioles. And also some goes to our pelvis and goes to our legs. And these are collected and eventually into the heart, especially right atrium. So let's take a look, distinguishing between arteries and veins. So artery has highest flow velocities, while veins are very low. And of course, arteries contain oxygenated blood so that it can feed throughout our body tissues and cells, while deoxygenated blood will be collected via vein system. So the color depends on the oxygen concentration of the hemoglobin inside red blood cell. And because of that, arterial blood is bright red, while venous blood is dark red. And you think about this artery has a very high pressure, while vein has very low pressure. So for the flow to happen, artery has a lot of pressure gradient, it's good, but vein do not. So instead of that, vein has a valve to be developed so that it prevents a backflow. Let's look at a little bit of morphological anatomy. The artery as internal diameter is narrower compared to vein. And one thing is, artery has to transcend pressures. So it has to endure that pressure. So one thing is thicker, while veins are thinner. And of course, because of that, the thick wall contains lots of muscles, and so it's more stronger than vein. But distensibility, which is how much it expands or swell, that capability, veins are higher. While arteries contain lots of smooth muscle cells and elastic tissue and fibrous tissue compared to vein, because it's a role of transmitting high pressure arterial blood throughout to the body as a pumping system. So let's get into more detail about the structure of these blood vessels. First, the most important artery wall. Let's see arterial wall looks like this, and this inside is where blood flows. So that's called lumen, and the lining of this wall is called endothelium. You can see here, and endothelium always sits on basement membrane. Okay, and so that's called intima. So it's inside, so it's tunica intima. And there's a middle part called tunica media and external part, tunica externa. And let's compare artery and vein one by one. So this left part is arterial blood vessel, right part is venous blood vessel. So tunica interna or intima. So this word, like we have three types, intima, media, adventitia. So that's how it works. And this tunica intima or interna contains endothelium cells. Both has endothelium, and endothelium should sit on the basement membrane. And while you can see this elastic lamina exists in the artery while not vein. So artery should be more elastic, vessel. And then you can see tunica media. So media contains smooth muscle. You can see artery has much more thicker smooth muscle cell because it has to endure this pressure, high pressure and high flow. And also there's another elastic lamina which doesn't exist in the vein. And adventitia, which is outside of the vessel, outer side part, contains a lot of collagen. So that's here. And the business end of this blood system is eventually a capillary where real exchange via diffusion is happening. And here is the simplest form of capillary. You can see one single layer of endothelium, and that sits on basement membrane. However, I have to say this is too simplified. This capillary also has a pericyte, which is involved in a lot of vein capillary functions. There's also sometimes smooth muscle cells at the arterial side and some part of the capillary. So in a summary, tunica intima or interna consists of not epithelium, but endothelium, because this is inside there's a float. Epithelium is usually outside, which is a single layer of squamous, like scale-like epithelium resting on a basement membrane. And media or middle layer is composed of, you can see, smooth muscle cells and also elastic fibers here. And the external part called tunica adventitia or externa consists of, you can see, there's a lot of collagen. Collagen producing cells are fibroblasts. And so this is collagen fibers. So now let's just review structure of vessel again. So artery, tunica intima contains endothelium that lines or the lumens of all vessels, and middle is the tunica media, smooth muscle cells, and elastic fibers. And the outer side of the vessel, tunica adventitia, contains collagen fibers. So you can look at the vessel as a kind of pipe, but floppy pipe or elastic pipe. And for the flow to happen, we need a driving force. In this case, fluid flow, pressure. Pressure is the driving force. So here, one side, P1, P2. If P1 is higher than P2, the flow will pass from P1 towards P2. And at the same time, this blood vessel is elastic. So inside the pressure is a vessel pressure, P sub V, while outside there's interstitial tissue. So interstitial tissue also has a pressure. So in order for the blood vessel not to collapse, which sometimes happens, this vascular pressure should be higher than outside. And so in terms of this vascular structure, we can define as a compliance, which means in terms of this delta P pressure gradient versus a volume changes. So if smaller pressure gradient, volume changes a lot, then we call more compliant. Okay, so aorta, which is the biggest artery, one of the biggest artery in our right after from outside of the heart, is aorta is very compliant in terms of it actually keeps the high pressure. So it's role as a pressure reservoir. While the biggest vein, which is getting into the heart from throughout our body, is called vena cava, and that is also very compliant, but that compliance comes from these changes in volume. So that's called, the role is a volume reservoir of our heart. So this vessel anatomy, as I simplified, but there are different types of artery. For example, elastic artery, it's a very big artery, is very elastic because from the heart, it actually transmits that high pressures throughout our body. So it has to be very elastic. And there comes muscular artery, and at the smaller arterial, and it goes down to capillary, and it collects as a vena and vein. And you can see endothelium is the same. There's a lot of elastic tissues in this artery side, and smooth muscles as well. And also fibrosis are much higher in artery. So next time we will see this part as more engineering point of view, vessel radius, resistance, and pressure drop. Thank you for your attention.